Am I audible? Hello? Hello?
Yes,
Okay, can you
yes, sir,
see?
you're on.
Okay, can you see my screen? Is PPT visible?
Yes, sir.
Okay. Okay. So today's topic is amino acid oxidation and production of urea. Okay. So talking about amino acid, we have already seen amino acid, right? How protein is made up of. So protein is basically made up of amino acid. This one amino acid, two amino acid, three, four, five, six, seven, eight, and they are joined together by the peptide bond. So this is a structure of the protein, right? Primary structure of the protein. So different amino acids are there, and they are joined together by the peptide bond. That is the basic structure of the protein. And now we have amino acid oxidation. So sometimes we want energy from this amino acid. So how to get the energy from amino acid? So what we can do with the amino acid? So we have this intracellular protein. Now this protein we can degrade it into the dissociate into individual amino acid. From dietary protein also we can get amino acid. So we have amino acid. So amino acid is basically made up of amino contain amino group and the carboxyl group and the side chain R group, and another one is a hydrogen atom. So this amino acid structure is there. So this amino acid structure can get dissociated into the ammonia plus carbon skeleton. Okay, so these are the two phases of this amino acid. So what this amino group can be used for? This can be used for biosynthesis of the amino acid, nucleotides, biological amines. So amino group is there. So this amino group can be used for the again for the synthesis of the different amino acids. So that is why the amino acid biosynthesis we have written here. Next, nucleotides. So there is under the nucleotides there are the purines and pyrimidines, the adenine, guanine, thymine, cytosine, uracil. Likewise, so for this nucleotide, if you see the structure of these nucleotides, purines and pyrimidines, there is a nitrogen atom involved in the backbone or in the side chain. So these nucleotides they can have this amino group as their biosynthesis for this their biosynthesis reaction. Next, biological amines. Next, biological amines can use this ammonia group. So these are the some of the fate of the this ammonia group. So next things, the utilizing reaction. So amino group we have got. We have made some use of this ammonia group. We have made the some biosynthesis of amino acids, some nucleotides we have made, some biological amines we have made. So these were some utility for things we have done. Now next, excess of ammonia is there, and we want to excrete that ammonia out of the body. Why we want to excrete this ammonia? So because this ammonia is very toxic for our body. So if ammonia toxicity is there, the ammonia accumulates in the brain, and their cerebral pressure increases, right? And then it may cause the death in the person. So that is why this ammonia is very dangerous. This ammonia, we want to get rid of this ammonia. So in order to get rid of this ammonia, we have this carbamyl phosphate. So this carbamyl phosphate can be formed from this ammonia, and this carbamyl phosphate will enter into this urea cycle. So it enters here. So it enters in this urea cycle, and ultimately the urea is being produced. Okay, so this is a urea is a nitrogen excretion product. So this urea is being produced here. So nitrogen excretion product. So this is a one phase of the amino group. Okay. So next phase we have whenever. So basically we are seeing the degradation of amino acid here. Amino acid degradation. So amino group we have get rid of in the either in the biosynthesis of other amino acid or in the form of the urea. Now next thing we have is the carbon skeleton remaining of this amino acid. So now how to get how to utilize this carbon skeleton? So this carbon skeleton can enter into the TCA cycle, citric acid cycle, as a alpha keto acid. So this carbon skeleton is getting converted into this alpha keto acid. Now this alpha keto acid is entering into this TCA cycle, and ultimately it is producing carbon dioxide, water, and some ATP. In this process, the oxaloacetate can be produced, and this oxaloacetate will be utilized via gluconeogenesis to produce the glucose. So this oxaloacetate, already we have seen the gluconeogenesis. The oxaloacetate is getting converted into the glucose, and this glucose will be the serving the need of energy for the body. So basically, this amino acid degradation involves urea cycle as a ammonia, getting rid of ammonia, and carbon skeleton as utilizing available carbon for other bodily functions like ATP production or for the glucose synthesis. So these are the two separate cycle, and today's lecture we will be discussing mainly urea cycle. TCA cycle already we have discussed, and there is a one bridge between them. So that is a aspartate arginosuccinate of citric acid cycle. So aspartate arginosuccinate shunt is there in the citric acid cycle. So this also we will be seeing in this lecture, right? So this is a basically flow of this lecture. So whole lecture we will be discussing this thing only. How so for example somebody is eating meat, the protein diet or eggs, high protein diet, and how this intracellular protein is going to break down into the ammonia component, carbon component, how this ammonia is going to go into the urea cycle, how urea is being produced, that we are going to see here. The carbon skeleton we will be discussing in bit about how this alpha keto acid enter into the TCA cycle, and we will be discussing about the aspartate arginosuccinate shunt of the citric acid cycle. So this is the flow of today's lecture. So metabolic fate of the amino group. So deposit the excess ammonia in mitochondria of hepatocyte. So metabolic fate. So we have amino group. So what happened with the amino group? So excess ammonia is there. How we can deposit this ammonia into the particular compound, or how we can get rid of this ammonia group, right? So this is a amino acid from the ingested protein. So this amino acid we are ingesting protein. Okay, so this is a amino acid is there. So amino acid is there, and what happened in the liver is that it is getting converted into the alpha keto acid. So this is a transamination reaction. So we have alpha one amino acid we have here, and alpha keto acid we have here. If you see other two substrate, alpha keto acid we have, then alpha ketoglutamate we have. So this amino group, what it will do, this is the amino group present in this amino acid. So this amino acid group, amino group transfer to this alpha keto acid, and this alpha keto glutarate. So this alpha keto glutarate get converted into this glutamate compound. Now you can see this is the ammonia here, and now this has attached to this alpha ketoglutarate. So there we have formed glutamate here. Okay, and once this ammonia group get removed, we have this alpha keto acid here. So this is COO minus C double bond O form here. R group is here. Okay, so this is basically alpha keto acid. So because of this, we call this as a transamination reaction. So amino group is transferring from one amino acid to another amino acid. So that is a transamination reaction. So now we have this alpha glutamate getting converted into the alpha glutamate. So these reactions are this transamination reaction is happening in the inside the liver. So this is the liver. So with the boundary we have seen that this is a liver. Okay. So glutamate is there. So glutamate is the emitting this ammonia group, and this ammonia group will be used in the synthesis of urea, uric acid, or it can be excreted as a ammonia. So you can see we have written something here, here and here. So what is this? So this is a ammonia. So ammonia. So we have taken the ammonia from one amino acid. We have transferred into the glutamate. Again we have removed the ammonia, and now this ammonia can be directly excreted out in some animal. We call this animal as a ammonotelic animal. Ammonotelic animals means these are aquatic. These include aquatic vertebrates, bony fishes, and the larva amphibians. So what happens is that in aquatic animals, once this ammonia is being ammonia can be directly excreted into the surrounding water, and it can get dissolved in the surrounding water. So that is a ammonotelic. So directly ammonia can be excreted. Whereas we vertebrates or in the animals, there is a this is a ureotelic animal. Okay, so ureotelic animal. So it means that we have to form the urea. So previous slide we have seen here, the urea is being formed. So this urea cycle we have to undergo this ammonia has to undergo the urea cycle, and this urea has to form. Okay, so this is a ureotelic. So terrestrial vertebrates and the sharks, they are ureotelic. Then uric acid. So in case of birds and reptile, there is a formation of the uric acid. So this compound is there. So uric acid. So this forming this animal as a excretion product of this ammonia. So that is a uricotelic. So there are different group based on how ammonia get excreted: ammonotelic, ureotelic, and the uricotelic. So these are the different forms in which the ammonia get excreted. So next we have alanine. So this alanine is coming from the muscle. So alanine is another amino acid. Again, if you put alpha ketoglutarate, this alanine will get converted into the pyruvate. Okay, so here double bond get attached, C double bond O, and here this ammonia group get transferred to the alpha ketoglutarate. That is the glutamate forms here. Okay, so that is so this reaction is there. Next, glutamine from the muscle and the other tissue. So what happens from the muscle and other tissue? You can get glutamine. This glutamine can either enter get converted into the glutamate and removes the ammonia. So basically, in the next slide, we are going to see how these reactions is happening. So the basic of this one is that during the amino acid catabolism, one amino acid is there. Our whole aim of this lecture is that how this amino acid is coming from here, one amino acid, how it is getting converted, transferred to the alpha ketoglutarate. Okay, it has get transformed now. Transferred. So that is a transamination reaction. Now glutamate has formed. Okay, glutamate has formed. Next, ammonia has get removed, and this ammonia can be excreted in the ammonotelic, urea, or uricotelic as a uric acid. Okay, and these are the different pathways which can supply the ammonia via different route. So gastrointestinal tract. So if you see the gastrointestinal tract, we have this our digestive system. So this is a gastric gland of the stomach. Parietal cells are there. These secrete the SCL. So this is our stomach. These are the parietal cells. So you can see here parietal cells. These secrete the SCL, hydrochloric acid. Chief cells secrete the pepsinogen. So pepsinogen is another enzyme which is a proenzyme. Proenzyme means it is not in the active form. Once it gets in the stomach, this pepsinogen can get converted into the pepsin molecule, and this pepsin molecule can act on our protein. So we have this protein chain, and it has to be broken down at the different places. For this one, we need the proteases. So this pepsin act as a protease, which will be the breaking the peptide our protein. So basically, we want to break our protein, then get the individual amino acid. So this pepsinogen helps in that process. Next, gastric mucosa. gastric mucosa secrete the gastrin. So this gastric mucosa is there, it secretes the gastrin. So here is there. So for example, you are eating some chicken or some meat, and now it is coming from your digestive system to here. Okay, so the meat has come here. So low pH, at the low pH, the degradation of meat is easy. So here low pH, at the low pH. Next we have pepsinogen enzyme. It is breaking down that meat portion. The protein is being broken down. Meat means the skeletal muscle or the muscles are mainly made up of the protein molecules. So these things are getting breakdown with the pepsin or the gastrin molecules. Or if you are if vegetarian diet, so the pulses on all those the protein, it will be broken down into the individual amino acid because of this pepsin and the chymotrypsin action. So there are different enzymes, pepsin, chymotrypsin, likewise, which will be helping in the breakdown of protein. So one point here is why this pepsinogen is in the inactive form proenzyme, because we don't want this gastric glands to get broken down. Okay, because the cells also made up of the protein and the cell membrane also contain protein. We don't want them to get hurt. We only want this inside portion to be available. The protein should act with the help of pepsin. So it is initially inactive, and at the end it gets active into the pepsin form in the stomach. So this stomach is there now. This protein particles go here. Here pH rises. This pancreas is there. Pancreas can secrete some enzymes. So pancreatic duct is there. Zymogen. Active proteases is there. So in the intestine you can see some active proteases get synthesized in the small intestine. So if you see the small intestine, the villus, intestinal mucosa absorb the amino acids. So now here you have eaten some meat particle or the pulses, and the protein present here, it has got degraded somewhat. So different fragment has produced. Again it comes into the small intestine. At the small intestine, this protein molecules get broken down into the individual amino acids, and now here is the this small intestinal membrane is there. It contains the villi of the small intestine. Small intestine. So villi of the small intestine. Here villus is there. Small intestinal mucosa absorbs the amino acid. So here amino acid can get absorbed. So these amino acids will come here and it get absorbed into this intestinal mucosa. And this is the structure of the pancreas. We have rough endoplasmic reticulum, zymogen granules, collecting ducts. We can see here. Okay. So these are this is how protein get inside the amino acid become available in our body, right? So here protein get degraded. Again, individual amino acid gets absorbed in the intestine. So this is a gastrointestinal tract. Next, transamination and aminotransferases or transaminases. So we have already seen this alpha ketoglutarate is getting converted. So this alpha ketoglutarate is there, and this L amino acid is there. So this L amino acid and alpha ketoglutarate. So we want to transfer this amino group to this alpha ketoglutarate via transaminase reaction. So with the transaminase reaction, we have transferred the amino group to this alpha ketoglutarate. Okay, so this is the transaminase reaction or the amino transferase reaction, and here alpha ketoacid form, here L glutamate form. And in this process, PLP is required. PLP is a pyridoxal phosphate. It is a coenzyme form of vitamin B6 or the pyridoxine. So again, we are discussing here the amino group, how amino group transfer from one amino acid to the keto backbone. Okay, so this is the keto backbone, and this is the amino acid. This is the keto acid. This amino group is getting converted, transferred to here. So here it is getting attached glutamate forms, and this alpha ketoglutarate get converted into the alpha keto acid. In this process, PLP pyridoxal phosphate is required. Vitamin B6 is a source of this pyridoxal phosphate. Vitamin B6 is also known as a pyridoxine. So that is why sometimes we say that along with our other essential nutrients like protein, fatty acid, carbohydrate, we require the vitamins. So this is a vitamins very critical for the this action. If this vitamins is missing, this transaminase reaction won't be processing further, right? So role of the vitamins in our diet. So this is one of the role of the vitamin B6 hydroxy. So glutamate is there. So now here we have found this glutamate. Okay, so this glutamate has formed. We have some amino acid. We have 20 amino acid. Out of 20 amino acid, any amino acid from any amino acid, we get this glutamate. Now in the next process, this glutamate is there, and we have glutamate dehydrogenase reaction. In this glutamate dehydrogenase reaction, what happens is that this glutamate get converted into this product, intermediate product. So this NAD or NADP plus can act as a intermediate in this one. So it get incorporated and we get NADPH. Okay, this energy molecule we get, and again if you hydrolyze it, this ammonia get removed. Okay, so and once we remove ammonia from this compound, so ammonia was attached here. So C double bond N double bond C in the first reaction. Once this ammonia get removed with the water hydrolysis reaction, we get alpha ketoglutarate. So basically, the glutamate dehydrogenase reaction is the reaction in which we are getting free ammonia molecule here. So this ammonia molecule we are getting in the second step. Ammonia transport in the form of glutamine. So So in our blood, the ammonia transfer form is in the glutamine form. Okay, so in the glutamine form, it can transfer. So L-glutamate is there. So how L-glutamate get converted into alpha glutamyl phosphate. Okay, so glutamine synthesis. So whenever we want to transport ammonia, we want to convert it into the L-glutamine. So how to do that? We have L-glutamate already. We saw how we can form the L-glutamate via transamination reaction. Next we have glutamine synthetase. So glutamine synthetase reaction is there. We want to form this compound. Okay. So glutamine synthetase. Okay. ATP is there. ATP incorporated. ATP removed, and we get gamma glutamyl phosphate. Okay. So here at this end we attach this phosphate group. Okay, so that is the gamma glutamyl phosphate. So if you measure from this alpha, beta, gamma, likewise we do denotation. So it is at the last, so gamma glutamyl phosphate. So again, this ammonia get incorporated here. So glutamate, if you see the structure of the glutamate, it is one ammonia group attached at the 1 to 2 number carbon. If you say glutamine, there is one more ammonia attached here. So here it should be ammonia. We want ammonia group attached. So ammonia NH4 plus we are adding here with the glutamine synthetase reaction, and inorganic phosphate we are removing. So now we have formed L-glutamine. So whenever you want to transport the ammonia from one location to another location, we have to undergo this reaction, glutamine synthetase. We have to attach ammonia to this glutamate with the help of the glutamine synthetase. During this reaction, this ATP is needed and this ammonia is needed. Next, glutamine. What happened at the liver mitochondria? So at the liver, for example, this ammonia glutamine goes to liver. Now we want to get rid of excess of ammonia. So this hydroxylation reaction is there, glutaminase, where hydrolysis reaction where this ammonia group get removed and this get converted into the urea molecule. So this is the formation of the L-glutamate here, and this ammonia molecule helps in the formation of the this urea molecule. So this is the one of the fate of the how this ammonia gets transported and how it forms the ammonia. Now glucose alanine cycle is there. So we have muscle and we have liver here. Muscle protein is there, it is getting degraded into the amino acid. If you are doing rigorous exercise, the muscle protein breakdown is happening, amino acid is forming, ammonia is forming, it is getting incorporated into the this glutamate form via transamination reaction. Now this glucose is there, via glycolysis we are getting pyruvate, and we have a lot of pyruvate. If it is anaerobic glycolysis, if muscle is stretching very fast, and this glutamate and pyruvate, it is forming the alanine and alpha ketoglutarate via alanine transaminase. So alanine transaminase ALT, alanine transaminase reaction is there. So here alanine has formed. Now this alanine transport from this muscle location into the blood. Now this alanine is transferred into blood. Now it goes from blood to the liver. Now it has come to liver. This alanine. We have alpha ketoglutarate. It is with the help of alanine aminotransferase. This is getting converted into the pyruvate and the glutamate. Okay. So pyruvate, so again transamination reaction, one amino acid is present on the alanine, which is getting attached with the alpha ketoglutarate, and we are getting the glutamate, and then the carbon skeleton is a pyruvate. This glutamate can undergo the ammonia conversion in the urea cycle, get converted into the urea. Next, this pyruvate undergoes the gluconeogenesis and it forms the glucose. This glucose can enter the blood and it can goes to muscle, and again it can facilitate the glycolysis. So this is the glucose alanine cycle we have. So already we have pyruvate available in the muscle. So this glucose alanine cycle can be useful here. Next, nitrogen excretion and the urea cycle. The cycle does span the two compartment. This urea cycle is there. So if you see this urea cycle, this is a urea cycle. So it happens in the liver. This urea synthesis happens in the liver. If you see this brown region, so this is a mitochondrial matrix of this liver cells. This is a mitochondrial matrix and this is a cytosolic portion. So cycle spans the two compartment. The cycle begins inside the liver mitochondria and three subsequent stages take place in the cytosol. So here is that cycle is beginning here. This glutamate is coming here, glutamate, then ammonia, carbonyl phosphate, then it is getting converted. So till carbonyl phosphate, this reaction is in the mitochondrial matrix. Then ornithine is coming and this it is joining with the carbonyl phosphate and we are getting citrulline. This reaction is also in the mitochondrial matrix. So this is the first step of the urea cycle, the synthesis of the citrulline. Next, citrulline comes out. It comes out in the cytosol. It is getting converted into citrulline AMP intermediate. Then it is converted into arginosuccinate, fumarate, and arginine. And then arginine is getting converted into the ornithine. The ornithine during this reaction, urea is being released. Okay, so these are the reactions. Okay, so one, two, this reaction 1, 2, 3, 4. These are happening here outside. So we call it as a 2A and 2B. So one complete reaction citrulline to arginosuccinate. So one reaction, 2, 3. So these three reactions occur outside, means in the cytosolic portion of the this liver cells, and this portion, this happen in the inside the mitochondrial portion of the liver cells. So this is a urea cycle. So we will see a little bit in detail the urea cycle, how urea has formed, right? So some of these stage already we have seen. Still we will go in detail. So we have seen glutamine. So glutamine, how glutamine form from the other amino acid, we have seen that with the glutamine synthesis reaction. Now this glutamine has from all location in the body. Now it has come from extrahepatic tissue. It came to liver. So it is traveling to the liver. Now it has come to liver. Now glutamine is entering into the mitochondria of the cells, liver cells. Now glutamine is there. With the glutaminase enzyme, it can get degraded into the glutamate and the ammonia. Okay, so it can get degraded into glutamate and ammonia. So this is one of the source of the ammonia. The ammonia is coming from the glutamine. Next source of ammonia is there, some amino acid is there, and now alpha ketoglutarate, alpha ketone acid, this transamination reaction is happening and we are getting glutamate directly at the liver location, and we are getting it converted into the glutamate. Again, with the glutamate we have seen the glutamate dehydrogenase reaction. Glutamate dehydrogenase reaction is a reaction in which we can remove the ammonia. So ammonia get removed here. So this is one of the reaction. Again, in the muscle, this alanine, glucose alanine cycle we have seen. Here glutamate can be formed. It can act as a source of the ammonia. So basically, here we are seeing the sources of the ammonia. Glutamine is one one source, glutamate is another source. Glutaminase is one enzyme, another is a glutamate dehydrogenase. And now we are getting ammonia here. Next we have oxaloacetate and aspartate. Again this aspartate to amino transferase reaction happening here. We are getting alpha ketoglutarate and the aspartate molecule. So aspartate amino transferase reaction. Okay, so till now we are getting this ammonia molecule here. So ammonia we have got here. This ammonia combined with this bicarbonate ion, this HCO3 minus, and with the help of 2 ATP, it is getting converted into the 2 ADP plus inorganic phosphate, and the reaction is catalyzed by carbamoyl phosphate synthetase. So this ammonia is there, so it is here. So this bicarbonate, so C double bond O, and this phosphate group is getting attached from this ATP molecule. So this phosphate, so ammonia, the carbon of the bicarbonate, and the phosphate from the ATP molecule. It has formed the carbamoyl phosphate molecule. Now this carbamoyl phosphate molecule inside the mitochondria, what happens is that it combines with this ornithine molecule. So ornithine molecule contains 1, 2, and these 3 5 carbons. 5 carbon are containing molecule, and 2 amino acids are 2 amino groups are present in this one. The ornithine combines with carbamoyl phosphate. So this ornithine combines with this, and now with the help of ornithine transcarbamylase, we are getting citrulline molecule. Okay, so citrulline molecule we are getting this carbonyl. This group is getting attached here. So 1, 2. Previously it was a 5 carbon. So this was a 5 carbon. Now it has a 5 carbon. This one, this group plus this group attached get attached here. Okay, so this is a citrulline formation. So this is a inside mitochondria reaction we have seen. So how inside the mitochondria of liver cell citrulline has formed. Okay, so this was about inside the mitochondria of the liver cell. Now we want to come outside the liver cell. Why? So the citrulline is entering into the cytosolic portion. So next we will go in the cytosolic portion. Let's see. So this citrulline, it has come out in the cytosol. So this was a liver matrix, mitochondrial matrix. Now it is a cytosol. In the cytosol, so from the mitochondrial matrix, the citrulline travels to the cytosol. Next, with the help of ATP, this arginosuccinate synthetase enzyme is there. Arginosuccinate enzyme, synthetase enzyme. If you put ATP and arginosuccinate synthetase enzyme, we will get citrulline AMP. So this is a citrulline molecule, and at this portion, this ATP's portion, AMP portion get attached. So this AMP portion get attached to here. Adenosine monophosphate get attached here. So this has attached with the previous citrulline molecule. We have got citrulline AMP intermediate. Okay, citrulline AMP, because this is adenosine monophosphate. Adenine is there, your pentose sugar is there, and one phosphate is there, that is a monophosphate. It get attached here. So this is a citrulline AMP intermediate. And in the next reaction, arginosuccinate synthetase reaction. Again, this is a part A of the reaction. Again, part B of this reaction is that during this reaction it requires arginine. So from where it requires aspartate. So we have to incorporate aspartate here. So this is aspartate molecule. If you attach aspartate to this portion, it get attached here, and this is a arginosuccinate molecule. Okay, so this is the arginosuccinate we formed here. So from where this aspartate came? This aspartate came from the previous reaction inside the mitochondria. The transamination reaction has formed the aspartate from the oxaloacetate, and this aspartate can also enter from this mitochondrial matrix into the cytosolic portion. So here it has entered. This is a arginosuccinate reaction. Next, arginosuccinase. The arginosuccinase is an enzyme in which this arginosuccinate, it get broken down into the arginine. So it is getting broken down into this arginine molecule. So from here it is getting broken down likewise, you can imagine. So arginine, and this fumarate portion is getting out here. So this is a fumarate portion. This arginine molecule, with the help of the arginase, during the hydrolysis reaction, it will remove this ammonia molecule. So this is arginine, we can remove this molecule here. So this is a urea is removed here. Okay, so urea is removed. When we say urea is removed, we have started with this reaction ammonia, and our aim is to remove the ammonia. So one ammonia it is you can see it is a blue in color, and another ammonia you can see aspartate also contain the ammonia, because aspartate is an amino acid. It is shown in the green color. And if you go in the back state still here, you can see it is a green color, so that is the ammonia we got from glutamate or the glutamine, and this blue ammonia we got from the this blue ammonia we got from glutamine or glutamate, and this green ammonia we got from the aspartate molecule. Okay, so this urea contains two amino group and C double one O group in the center. So this is arginase reaction. We got rid of the two ammonia. So this urea, it enters the blood and it can be filtered by the kidney and excreted out of the body. So this is how the urea is formed. Next, this arginine getting converted into ornithine. Ornithine again enter this cycle. So this is how urea cycle works. How the ammonia group get converted into the urea molecule. The citric acids and the urea cycle can be linked. So whenever we have to link something, we have to have something common in between them, right? So the fumarate is produced in the organosuccinate reaction is also intermediate in the citric acid cycle. So here we can see this fumarate is there. So if you know the citric acid cycle, TCA cycle, you know there is a synthesis of the fumarate in this cycle. This fumarate can be utilized. So this fumarate can get converted into the again into the malate and then oxaloacetate, and it can be entered into the gluconeogenesis or it can get broken down in the TCA cycle, right? So this fumarate is produced in the reaction. Arganosuccinase reaction can be used as an intermediate in the citric acid cycle. So sometimes people dub it as a Krebs bicycle. The aspartate found in the mitochondria via TCA cycle serves as a nitrogen donor in the uh, urea cycle. So this aspartate can be formed from this TCA cycle via transamination reaction with the oxaloacetate. So this aspartate can get oxaloacetate can undergo transamination reaction to the aspartate and it can act as a nitrogen donor. So already this is what we have discussed. So, in short, if you are talking about urea cycle, we are starting from here: glutamine, glutamate. Okay, ammonia group. Okay, our aim is to remove this thing. Okay, next from here, oxalosuccinate is there. This oxalosuccinate can be from the TCA cycle. So that is the linking up to your T cycle. You will see a little bit further again. But okay, so transamination reaction is there. Glutamate, it has transferred its amino acid to here, to oxaloacetate. Now we have got amino group here. So this aspartate molecule, okay, it also contains ammonia. We don't want it. This ammonia, we don't want it. So we process this in the urea cycle. So this ornithine to citrulline, citrulline to arginosuccinate to fumarate and arginine to ornithine. Okay, here we got urea cycle, urea. Okay, urea got whether ammonia removed. This is a blue ammonia. Okay, blue is there. Whether it is green ammonia from aspartate. Okay, it is there. So these two things we have got removed. So that is how we get rid of ammonia. So the pathway interconnects reduce the energetic cost. So if you connect the TCA cycle and the urea cycle, basically it is reducing the energetic cost of the urea cycle. The fumarate generated in the urea cycle is converted into the malate. So the urea cycle, the fumarate is generated, it gets converted into the malate, and then the malate is transported into the mitochondria. Again, you know the fate of the malate. It can get converted into the oxaloacetate, and this oxaloacetate into the citric acid cycle. It is being utilized. Basically, the malate can be utilized in the TCA cycle. Inside the mitochondrial matrix, ATP is generated in the malate dehydrogenase reaction. Okay, so with the help of this malate dehydrogenase, NADH is generated. We know that each NADH molecule can give the 2.5 ATP during the mitochondrial respiration. So during the mitochondrial respiration, 2.5 ATP can be generated, and this serves as a source of energy. Otherwise, if you don't have this fumarate as a intermediate, what how much energy cost is involved? So if you see here, two ATP is involved. This two ATP here, one ATP is involved. Again, one ATP is involved in this remote. So total four ATP will be involved in the expenditure will be involved. So if you have connection, we can have some energy saving. We can generate 2.5 ATP during this process, right? So coming to the link between the TCA cycle and urea cycle. So urea cycle, the whole lecture we are discussing is the urea cycle. So this is a liver. So this carbonyl phosphate has formed, ornithine and carbonyl phosphate come together. Okay, citrulline has formed. Citrulline, it has undergone to the arginine succinate synthesis. Okay, next arginine has formed. Arginine has given the ornithine and now urea has removed. Okay, so this is removed. But what happens is that this arginosuccinate can give us this oxaloacetate in this reaction. So during this reaction fumarate is also formed, right? Arginosuccinate fumarate. Okay, this fumarate has formed. Now can convert into malate. Malate here it enters. Malate can give the energy in the form with the help of malate dehydrogenase. So here NADH we got. So the NADH is around 2.5 ATP molecules we got here. So it enter the TCA cycle. Likewise fumarate, malate. Likewise. So fumarate directly doesn't enter. It gets converted into malate. The malate enters into the TCA cycle. Then again oxaloacetate we get. Okay. Can TCA cycle help with the urea cycle some way? Okay. Oxaloacetate we have. The glutamate, if you have glutamate contain amino group. Okay. You transfer this amino group to this oxalate molecule. And this oxalate molecule get converted into the aspartate. Now aspartate is there, and this aspartate can get converted for this citrullyl AMP to form the arginine succinate. We need the aspartate, and this aspartate will be also helping as a one of the component of the urea nitrogen. Okay, NH2 component. So arginosuccinate can be formed here, and then again it gets converted to the oxaloacetate with the glutamate, alpha glutamate can be formed, and this outside in the cytosol, this oxaloacetate can get converted into the malate also. So this is a link between the TCA cycle and the urea cycle. Okay, so this is our TCA cycle, this is our urea cycle, and these are the some of the steps which are happening inside or the outside the mitochondrial matrix or the cytosol. So this is the cytosolic portion, this is a mitochondrial portion. So this is a link between the TCA cycle and the urea cycle. The synthesis of N-acetyl glutamate and its activation of the carbonyl phosphate. So here we have seen during the activation ammonia to carbonyl phosphate, this carbonyl phosphate is synthesis needed. So here we are seeing the activation. This carbonyl bicarbonate and ammonia is combining, and we are getting the carbonyl phosphate here. During this reaction, this carbonyl phosphate synthetase 1 is involved, and this what it does, the 2 ATP get converted to ADP and inorganic phosphate. And during this reaction, N-acetyl glutamate. So this is a glutamate molecule, and this is a acetyl group attached here. So N-acetyl glutamate act as an activator of this carbonyl phosphate synthetase. Okay, synthetase 1. So how to form it? Acetyl CoA combined with glutamate. In the N-acetyl glutamate synthesis, this CoA get removed and we get N-acetyl glutamate. It says as a activator. So this reaction get activated here. So this is the activation of the carbonyl phosphate synthetase 1 with the N-acetyl glutamate molecule. So if some genetic defect is there, can we treat the patient with a genetic defect in urea cycle? So the basic whole topic, whole lecture we are talking is that the ammonia is being formed in the body, and we want to get rid of the ammonia. So some people is there who have that defect in this urea cycle. So if defect in urea cycle is that, it means that they cannot remove the ammonia from the body. So ammonia can get excess in the body, and again we have, as we have discussed, the osmotic pressure in the brain can increase, and it can be a fatal for that individual. So we want to remove ammonia. So how to treat that patient? So we can give benzoate or phenylbenzoate to this patient. So let's see what is happening with the benzoate. So this is a benzoate molecule plus CoASH. With the ATP molecule, it is getting converted into the benzoyl CoA. So benzoate, CoASH and ATP, we are getting benzoyl CoA. Okay. So benzoyl CoA we have got. So still no ammonia. Next, glycine is there. So okay, here ammonia is there. In the ammonia amino acid glycine, it is getting attached with this benzoyl CoA. Here we are got benzyl glycine. So benzyl. So here this glycine molecule get attached with this benzyl CoA. So CoA get removed, CoA SH, and we get hippurate. So because of this hippurate, you see the structure of the hippurate. We have benzyl structure and the glycine group attached with it. So that is a benzyl glycine, and during this reaction we can remove some of the ammonia. So you can see ammonia can be removed. So if you give patient the benzoate, it get converted into benzyl CoA and then attaches the amino acid glycine with it, and we get hippurate, and this hippurate can be excreted in the urine. Okay, so in the urine this benzyl hippurate can be excreted. Likewise, this treatment involves phenyl butyrate. So this is the phenyl butyrate. It undergoes the beta oxidation into the we get acetyl CoA and the phenyl acetate. So this is getting beta oxidation. Now here we have phenyl acetate. Again with the ATP getting converted into AMP and the this PPI, and this phenyl acetate is getting converted into phenyl acetyl CoA. Okay, so phenyl acetyl here with the phenyl acetate we are writing acetyl CoA SH. So phenyl acetyl CoA we have got. So like benzyl CoA, now we have phenyl acetyl CoA. Next we need amino acid. Which is this amino acid? Glutamine. Okay, glutamine contains some amino group. Here is amino. Here is amino. So some amino acid ammonia can get removed, right? So here CoA SH got out, and here phenyl acetyl glutamine. So here we are losing glutamine or the glycine in this reaction with the phenyl acetyl glutamine or the hippurate. But if you see, the hippurate and the phenyl acetyl glutamine are comparatively non-toxic, and they can be excreted in the urine. So this is how people try to correct the genetic defect in the urea cycle as a treatment, one of the treatment, right? So we have this amino acid. So we are talking a lot of amino acids. So just a refresher, we have is a non-essential amino acid. We don't require this in the diet. Uh, Body can synthesize them: the alanine, aspartate, asparagine, then glutamate, and the serine. Then conditionally, sometimes it is essential is that in case of human, arginine, cysteine, glutamine, glycine, proline, and tyrosine. And if it is essential, it is histidine, isoleucine, leucine, lysine, methionine, phenylalanine, threonine, tryptophan, and the valine. So these are some of the amino acids whose fate we are discussing here. So amino acid fate, till now we have discussed the fate of the amino group, how this amino group is getting converted into ammonia, and this ammonia is entering into the urea cycle, how it is the urea cycle, in the inside urea cycle this urea is being formed. So that was the about urea cycle. Next, carbon skeleton. So at the start we saw ammonia and the carbon skeleton. Let's talk about the carbon skeleton in short. So pathway for the amino acid degradation. So this arginine is there, glutamine is there, histidine, and proline. So arginine, glutamine, histidine, proline can undergo the conversion into the glutamate. This glutamate can undergo the conversion into the alpha ketoglutarate. If you see, alpha glutarate is a part of the TCA cycle. Next, so all amino acids are coming ultimately into the TCA cycle, their carbon skeleton. Okay, let's see the carbon skeleton of the isoleucine, methionine, threonine, and valine. They are getting converted into the succinyl CoA. Succinyl CoA getting converted into succinate, then fumarate. Here, phenylalanine and tyrosine skeleton is coming into the form of the fumarate. Aspartate, asparagine coming in the form of the oxaloacetate here. Alanine, cysteine, glycine, serine, threonine, tryptophan, their skeleton is getting converted into the pyruvate. Isoleucine, leucine, threonine, tryptophan, their carbon skeleton is getting converted into the acetyl CoA. Leucine, lysine, phenylalanine, tryptophan, tyrosine, their carbon skeleton is getting converted into the acetoacetyl CoA. Okay. Okay. So these are the fate of the carbon skeleton. So we have two colors here. If you see here, so this is a somewhat brown color and this is a blue color. So we have seen leucine, lysine, phenylalanine, tryptophan, tyrosine. and the isoleucine leucine threonine tryptophan in the blue color. Why it is in the blue color? You see here it is a ketogenic. Ketogenic means it is getting converted into the acetyl CoA. Finally, so these amino acids also get converted into acetyl CoA, acetyl CoA, and this acetyl CoA can serve as a molecule which helps in the synthesis of the ketone bodies. Okay, acetone, beta hydroxybutyrate, acetoacetate. This can be formed as a ketone bodies from this acetyl CoA molecule. So that is why some amino acid we call it as them as a ketogenic, and some amino acid these are the glucogenic. Why we call it as a glucogenic? Because this glutamate is there, alpha glutamate forms succinyl CoA, succinyl fumarate, oxaloacetate. From this oxaloacetate, it can undergo the gluconeogenesis and the glucose can be formed. So that is why some amino acid we call them as a glucogenic and some amino acid as a ketogenic, and some are the both glucogenic and the ketogenic. Let's search if such amino acid is there. Threonine is there, it is a ketogenic. Threonine is there, it is also glucogenic. So this is both ketogenic and the glucogenic. Tryptophan is a ketogenic. Tryptophan is also glucogenic. Leucine, can you see leucine here? Tyrosine, phenylene. So leucine is lacking. Isoleucine. So isoleucine is here. Isoleucine is there. So both ketogenic and glucogenic. Let's see whether we have phenylalanine. Phenylalanine we have here. Phenylalanine we have here. Tryptophan we have here. Tryptophan we have here with the glucogenic. Tyrosine we have here. Tyrosine we have here. So both ketogenic and glucogenic. Let's see lysine. Lysine, 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 no lysine. So lysine is a strictly ketogenic. Leucine, 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 leucine. So this is also a strictly ketogenic. So that is how the amino acid gets their name as a glucogenic, ketogenic, or both ketogenic and the glucogenic, right? Leucine and lysine we can see from here is a strictly ketogenic. Some amino acid these blue are the both blue are ketogenic, but some of them can be both. Okay, so that is how the pathway of amino acid degradation happens. So during this pathway amino acid degradation, we need some cofactors to help transport the one carbon atom. In that carbon, this biotin molecule can help. Then this tetrahydrofolate molecule can help, which is 6-methylpterine, methylpterine, N-methyl benzoate. It contains in its structure, and the glutamate contain. Then 5-adenosine, yes, adenosine methionine, idomate helps. So this biotin, again this amino acid, these vitamins or folate. So sometimes people say the folic acid is necessary in the diet. So this is involved in the one carbon transfer in this process of amino acid degradation, right? So some branch amino acid are there like valine, isoleucine, leucine. With the help of branch chain amino transferases, it get converted into the keto acid, and with the help of branch chain alpha keto acid dehydrogenase, it get converted into acyl CoA derivative. If this enzyme is deficient, we get a maple syrup urine disease, right? Degradation of asparagine and aspartate. So this asparagine amino acid is there. With the help of asparaginase, it is getting. So just one amino acid as an example we are showing right now. Asparagine amino acid with the asparaginase, ammonia get removed during hydrolysis reaction. We get aspartate here. Aspartate aminotransferase get converted into the oxaloacetate. So here amino group transfer to the alpha ketogenic, we get glutamate and the oxaloacetate here. So this oxaloacetate can enter the TCA cycle. The 20 common amino acid after losing their nitrogen atom are degraded by dehydrogenation, decarboxylation reaction to yield the proportion of the carboxyl. So that is why in your syllabus we have this decarboxylation reaction, deamination or the transamination reaction or the dehydrogenation reaction. So whenever we say you will see the word transamination, why it is in the syllabus. So this is the reason. We have to get the rid of ammonia. We need ammonia back again our body. So that is why these things are there: decarboxylation, dehydrogenation, or the transformation. So those portions are degraded to, so carbon skeleton, it can degrade to acyl CoA and then completely oxidize to the carbon dioxide and water and generate the ATP. So whenever we say this here, so acetyl CoA has formed. So it can enter into the TCA cycle, and we know in the TCA cycle we get carbon dioxide and water, and during this process ATP is generated, right? So that is what is mentioned here by this oxidative phosphorylation in the mitochondria, right? So that's it for today's lecture. But still one more point.